Okay, now we're going to move on to multi-gene traits. So in this situation, we're going to deal with the situation where multiple genes can affect the same character. And so to start out, we're going to talk about skin coloration in corn snakes. So corn snakes can have 4 colors: camouflaged, black, orange, and albino. So these are pictures of the different colors, and researchers have determined the genotype of these. So it's important to remember that this is a single character. This is a reminder for what a character is. You should re-watch the lecture from chapter 3 if there's any sort of confusion. But the color of the snake represents one character. But in this case, we have 4 different traits. So there's 4 different versions that this color can take. And so this is unusual compared to the previous chapter. We had dealt with dihybrid crosses before, but in that case, it's important to remember that there was 2 different characters that we were looking at. We were looking at both seed shape and seed color. In this case, we're only looking at 1 character. But the genetics is very similar. So there's 2 different genes, and so this example, we're following the Drosophila kind of approach, where the wild type is listed with a plus sign. And so there's 2 different genes to worry about. So one is O, one is B. There's 2 alleles of each, and these again are completely dominant to each other. So when we speak of dominance, we're saying that the O plus is dominant to the little o. We also are going to see this kind of methodology used a lot for multi-gene traits, where the way you specify this dominance relationship is by using this dash. And so how do you interpret this genotype right here? O plus over minus, B plus over minus. What this means is that the following are the same. So this genotype is the same as this genotype. So as long as there's 1 O plus allele, it doesn't matter what that second allele is. So it can either be O plus or O plus O. And the same is true with this one right here. So there's actually 4 different genotype combinations that lead to this camouflage color. And I'll leave it to you to work out the other 2. In terms of the black color, you have to be homozygous recessive for the O gene, but then for the B gene, you just have to have, again, a single copy of the B plus allele. So it doesn't matter what this one is. So in this case, there's 2 genotypes that actually would lead to a black color, a black trait. Orange, we flip that. So as long as the B gene is homozygous recessive and the O gene has 1 dominant copy, then the color of the snake is orange. And then albino is a situation where you're homozygous recessive for both. So let's say we take an individual that's camouflaged and we cross it to an individual that's albino, and kind of in the style of a dihybrid cross, we further require that both of these individuals be true. So in this case, I have an individual that's homozygous crossed to an individual that's homozygous. And so let's look into the F1 generation. The F1 generation would now be heterozygote for both, O plus over O, B plus over B, and this individual would be camouflage color, just as a reminder, because of this tells you that it would be camouflage right here. So O plus O, B plus B would fit within that category. So what happens now if I intercross these individuals? So again, this individual can produce 4 different gametes. And then if I cross it to another individual with the same genotype, you would again have the same set. So what you would have is a 4 by 4 square. And then this is almost exactly the same as a dihybrid cross. And so you would again group anything that had the same phenotype. So anything that shared a phenotype could be grouped together. And then if you go through that whole process, you will come out with the familiar ratio of 9 to 3 to 3 to 1. So if you crossed a camouflage to an albino snake, you would get a camouflage snake. If you intercross the brothers and sisters, you would get a range of all 4 colors, and you would get the ratio of 9 to 3 to 3 to 1. So this is just a simple example of how 2 genes can control a single character, resulting in 4 different colors, 4 different traits, and kind of the expected ratios from that. So now we're going to move on and make things a little bit more complicated. So everything about the approach before is similar with the exception of the existence of epistasis. And so epistasis, so 2 genes still affect a trait. Typically dominance and recessiveness still applies, but now we allow 1 gene to mask the phenotypic effect of another gene. So what is an example of epistasis? So I own a chocolate Labrador. I love dogs, so I like to use this as an example. Anyone that knows Labrador retrievers know there's 3 primary colors that they come in. So you can get a black lab, a chocolate lab, and a yellow lab. And breeders have gone to the trouble to figure out what is the genetic basis of this. And so why would they do that? Well, they want to have the ability to produce litters in certain combinations. So they might know a bunch of people want 4 people want a black lab puppy, 4 people want a chocolate lab puppy, so they want to set up a cross where they know they can have a mixture of both within those litters. And so what they found was that it was controlled by 2 different genes, so very similar to the snakes. And again, we're kind of following this approach where we indicate dominance and recessiveness. So, they've gone through the trouble to figure out what is the genotype-phenotype relationship. And what they found was that if you have a dominant allele of B and a dominant allele of E, then you are going to be a black lab. And so the second allele doesn't really matter. So it can be big B, big B, big B, big E, little e. That's a totally fine genotype for a black lab. If you're homozygous recessive for the little b gene, they found that they're actually going to be brown. So it doesn't matter what your second one was, but as long as you had homozygous recessive for B and 1 copy of the E, then you're a chocolate lab. So far, this just looks like one of the categories of the snakes. But what is surprising is that they found that if you were little e for both, basically that meant that you're a yellow lab no matter what. And so that, in this case, it does not matter what this gene does. All right, so let's take these rules and let's just do a standard dihybrid cross. So let's assume that we've crossed a black lab, a true breeding black lab, to a true breeding yellow lab. And as a result, we have a heterozygote for both genes. So big B, little e. And so we know, based upon this rule right here, that that individual dog would be black in color. And we crossed it to another individual of the same genotype. And then what sort of individuals would you expect? So again, we do a Punnett square. This individual can produce 4 different gametes. This male dog can produce 4 gametes. This female dog can produce 4 gametes, so we list them all on the sides of the square, and now we just fill in the 16 boxes inside. So we just combine these. These are the genotypes, and now we've also gone to the trouble of filling in the color based upon this rule right here. And so now we group these all together, and we get a 9 to 3 to 4 ratio. Okay, so because this is called epistasis because in effect what it's doing is it's masking one of the expected phenotypic classes, one of the expected trait classes. So in the snakes, we saw a 9 to 3 to 3 to 1 ratio, but here we're seeing a 9 to 3 to 4 ratio. And why is that? Well, we still have kind of this 1-to-1 relationship, But now both of these categories, these genotypic categories that were separate, are both producing the same color. So all of these genotypes produce the same color, and so we add this 3 and the 1 together to get the ratio of the 9 to the 3 to the 4. So it's recognized by the fact that we have one less, so we still see these like 9 to 16, these 16 ratios, but now we have one less trait that we expect, so we would expect it to see 4 different traits, and now our ratios are modified as well. So this is the definition of epistasis. So you have to have a trait that's controlled by 2 genes, but instead of seeing a 9 to 3 to 3 to 1 ratio, what you're seeing is a skewed ratio with less classes. The second example of this is squash fruit color, but the relationship is a little bit different. And so again, you should get used to being able to interpret this to figure out the color of the different classes. So the colors of these squash can either be white, green, or yellow, and there's 2 genes that control it, and these are the rules that tell you. So this class looks a lot like the black lab. If you have a dominant allele of both, you're white. But now all of a sudden, this class right here is also white, kind of unexpectedly. So if you're little g, if you're homozygous recessive, then you're also white. If you have one dominant allele of the big G, you're homozygous recessive, then you're green, and then if you're homozygous recessive for both, then you're yellow. So again, we've masked one of the expected traits, and we only see 3 different colors instead of the expected 4. So this again is an example of epistasis, but what you should note is that the ratios are going to be a little bit different. So the genotypes that are grouped together are different, and so as a result, instead of having a 9 to 3 to 4 ratio, you have a 12 to 3 to 1 ratio, which is determined by these rules right here. So this also motivates the definition of different types of epistasis. So 2 common forms of epistasis are recessive epistasis and dominant epistasis. So this means the recessive alleles at one locus, one locus, mask an allele at a second locus. So Labradors are an example of recessive epistasis. And so what does that mean? Well, if you have the recessive alleles of this gene right here, it doesn't matter what your B combination is. There's no combination of B that can affect the color of this dog, and so that's why it's called recessive. So because this, in the recessive form, masks the effects of this gene, it's recessive epistasis. The squash are an example of dominant epistasis. So as long as you have a dominant allele of the W gene, it does not matter at all what your genotype is of the G gene. All of the 3 combinations of genotypes are all the same. And so again, this is an example of masking the effect of a second gene. And this is called dominant epistasis. And so these are typically just recognized by the ratio. So if you see a 9 to 3 to 4 ratio, then you're going to expect that it's probably recessive epistasis. If you see 12 to 3 to 1 ratio, then this is an example of dominant epistasis. And you can actually even get more, more complicated than that. So, these are 8 different cases of traits that are controlled by 2 different genes. And so these are different ways that you can modify the 9 to 3 to 3 to 1 ratio. So these first 2 are an example of recessive epistasis and dominant epistasis. But there's also examples for grouping additional ones of these genotypes together. So this P flower color, you grouped these 3 genotype classes together because they all produce the same color. And so you add up the 3 plus 3 plus 1, and so you end up with a 9 to 7 to 1 ratio. Or you can group the 2 middle ones together and have the same phenotype. So fruit shape and squashing is an example of that. And so you end up with a 9 to 6 to 1 ratio. So there's all sorts of different groupings that can occur. And just the details, you have to know the molecular kind of details and the cellular details of the trait to understand how this arises. But the thing that's important right now is just to know that all of these are forms of epistasis. And they're all recognized because they have less less trait categories than expected, and they also don't follow the typical 9 to 3 to 3 to 1 ratio. All right, so this is the end of the section on epistasis.